So when we're looking at proteins, we talked about proteins, right? Everybody remember that? And our objective here is to understand this idea of what is an antibody, right? Because when we're talking about ELISA, one of the core concepts is there's a few core concepts. One is an antibody. Two, you have the surface proteins. So we have ELISA, antibody, surface proteins, and then you have T cells. Well, in a normal immune reaction. Now we're not going to be dealing with T cells in an ELISA, but in a normal immune reaction in your body, what happens is the antibody sticks to the tumor cell in this case, and that tumor cell, the antibody sticks to the tumor cell and then also acts like a flag to the T cell. The T cell is going to engulf and kill the tumor cell. That's what should happen. Now, this is very difficult to happen in your body. And in fact, recently, the latest Nobel Prize went to a group of scientists that discovered that when your T cells are first made, they go through a process of recognizing. They go into the thymus. It's an organ in your body. It's a gland. They go into your thymus and they get trained. That's where your white blood cells are trained to recognize you. And how do you do that? Well, what happens is when the T cells get to, or when the white blood cells get to the thymus, if they stick, so there's an if then. If they stick to your tissues, they're killed. Then they are killed. If they stick, then they are killed. So the only the only white blood cells that get out of the thymus when they're first made are cells that don't stick to you. Does that make sense? That don't react to you. You don't want your immune cells reacting to your own body, because then your cells start killing your own body. These are called autoimmune diseases. And some of these diseases are things like, you may have heard of them. There's MS, which is multiple sclerosis, okay? You might have heard of, has anybody heard of that, MS?
Yeah.
Yeah, so that's, my brother died of MS, but it also happens much more in women than it does in men, but my brother got it, and we think it's triggered by a viral infection early on in life. Herpes virus infection, which could be a cold sore.
Or like shingles.
Or shingles, yes. All these herpes are a family of viruses, not just the sexually transmitted disease that a lot of people think about, okay? But we think it might be Epstein-Barr virus specifically is what they think might be a trigger for developing MS later in life. Another one you might have heard of is lupus. I'm not sure how to spell that. Probably U.S. All right. Lupus. You probably have heard of asthma.
what'd you say was might be a trigger for later in life?
Epstein-Barr is the name of the virus.
Okay.
It's a herpes virus. It's in the family of herpes viruses. There's an MS, there's lupus, there's allergies. Okay, there are what else? I just said it a minute ago. Asthma, right? So in any case, there's a lot of diseases that occur because your body is attacking, your own body attacks itself. These are when you have an allergy, your immune system's overreacting. And there's even we have a student in the building that has a cold allergy. When he goes outside and it's cold, his body starts reacting.
On our floor?
Huh?
On our floor?
I can't hear you.
On our floor?
Yeah, he's on our floor. He goes out in the cold. He has to wrap up extra in the wintertime. When we went to Florida, he couldn't go in the ocean because it was too cold for him. He might react while he's in the ocean. And we're talking about swelling up. We're talking about the entire immune reaction happening because of cold. In fact, a roommate of mine in med school also had this. So in any case, that's the normal. That's what's happening now. We're going to use this. We're going to use a part of this process, this recognition of the great thing is that this.
Charge that, Mr. Mendoza. You're at 13%, 12 now.
Oh my goodness. Well, we're going to have to find it. Yeah, let me pause this and get an extension. All right, so the key is that these antibodies are sticky. Everything's about shape, and stickiness. All right. And now you see what. By the way, just to clarify this illustration, when the T cell gets near the tumor cell, and it has it's recognized through the antibodies. Okay. Then what happens is this T cell releases cytotoxic chemicals, little poisons, and it kills the cancer cell. Okay.
You say cytotoxin?
Cytotoxin. So cyto means cell, and toxin means it's toxic.
I know. Mr. Greasehammer told us. He
Oh,
gives
okay.
us the root of every word.
Oh, good. That's excellent. That's excellent. So when we talk about ELISA, though, it's called an enzyme-linked immunoabsorbent assay. So you have enzyme-linked immunoabsorbent assay.
Is immunoabsorbent one word or two?
There should be an S in it, unfortunately. I have to erase that.
Oh, that's what ELISA
It's one
stands for?
word. It's one word, first of all.
Is that
But
what ELISA stands for?
that's what ELISA stands for, yeah. I was very messy there. I apologize. Here's English again. Absorbent.
There's no A?
There's no A. Isn't that annoying?
That's so stupid.
It's so annoying. Immunosorbent.
Wait,
Sorbent.
immunosorbent?
Yeah.
It's not absorbent?
That's what it.
Oh, so
Just
I gotta
let's go
erase
with
it?
what it is. That's how you spell it. I'm not even going to try to defend it. It is what it is.
Wait, so they have... Wait, what?
That's where it comes. That's why it's always confusing to me when I think of ELISA, because I always forget there's no A. So
Or
it's
B.
immunosorbent.
There's no AB.
So it's immunosorbent assay. So ELISA, ELISA. And the job of ELISA is to detect and quantify. Now, so I'm just using these words. I'm going to write them down. Then I'm going to go back and we're going to talk about what they mean. They're used and they're going to quantify specific proteins, peptides, antibodies, or hormones. In a biological sample. So let's go back now and let's kind of break this down. So first, let's talk about this word enzyme. So just to be clear, enzyme is usually a protein. It can be RNA as well. And the whole job of an enzyme, and those of you that had me in 9th grade last year know this, but an enzyme, what it does is it takes a substrate, we call it a substrate. It's basically a molecule or more than one molecule, right? So it could be two molecules being put together, or it could be one molecule being broken up. But it's some kind of molecule. And that molecule has to have a shape that fits in what we call the active site of the enzyme. Now you could think of the active site as the mouth of the enzyme, okay? And so this is an enzyme. Now, normally, a substrate, you'll have a reaction. So the substrate, another word for a substrate is a reactant. In any chemical reaction, you have a reactant that then leads to a product. In a chemical reaction, this is a chemical reaction.
Leads to what?
A product.
I thought that was an A, not a P.
Oh, well, I think it's clearly a P, but
I'm sorry.
No, it's not your fault. I also, it can be very messy. Let me redo it. Product. Is that clear?
Sorbent.
Yeah.
Product. And so the product is essentially different from the reactant. The atoms have been rearranged. That's what a chemical reaction does. It rearranges the atoms in a molecule. So it does not make new molecules. Remember, that's the first law of thermodynamics. We can't create or destroy any matter or energy, right? Yeah, I can actually zoom in a lot. So when we're dealing with a reactant, it's going to make a product. It's going to be different, and we can take one thing and make them into two things. So that plus, let's say this. So we started with this reactant, and we ended up with these two products. We're literally breaking it up. So the reactant, when we're dealing with enzymes, we call the reactant the substrate. That's the molecule that binds in the active site, in the mouth of the enzyme. Then it leads to a product. Now, a good example of this, I'm going to write it up here, H2O2. Some of you might have it at home. It's called hydrogen peroxide. Will break down, especially it will break down into H2O and oxygen gas. And when oxygen gas is produced, it bubbles. So if you put this H2O in a test tube or a container, you'll see maybe one or two, maybe a couple bubbles. That's the H2O breaking down, H2O2, hydrogen peroxide, that's the name of it. Hydrogen peroxide.
Why does biology have all those weird letters like X's and Q's and Y's.
Oh, we have every letter there is. Hydrogen peroxide. Now, you've used that. You probably have it at home, hydrogen peroxide. You also produce hydrogen peroxide in your body when you go through metabolism. This is dangerous because hydrogen peroxide is a poison. That's why we pour it on our cuts, because we want to kill the bacteria. And you'll see when you pour it on your cut in your blood, you'll see your hand bubble. Have you ever had that experience? That's because you're producing oxygen. Now, this is killing the bacteria. It's also killing some of your cells, but it's mainly killing the bacteria. Now, if you have it in a bottle, there's just a few little bubbles, but once you pour it on blood, you get a lot of bubbles really fast. And that's because you have this enzyme. And so what does an enzyme do? So let's take a look at a graph. You know, I love graphs. And if we look at a graph, whenever you have a chemical reaction, the substrate or the reactants start at a certain amount of energy. And over time, in a reaction, when a chemical reaction happens, if you give it enough energy, if you give hydrogen peroxide enough energy, it'll react. It'll start to change. And once it starts to change, it gets the molecules become low energy. They go from high energy, it's high here, to low. The universe is lazy. Everything wants to get to the lowest energy state, naturally. But it's hard for this reaction to happen. You need to have this amount of energy to happen for it to occur. A good example of this happening is paper burning. You're writing on paper, are you not?
No.
Why isn't it burning right now?
Because it's not hot enough.
There's not enough energy. That's right. You're right. It's not hot enough. It's not enough energy. So I can take a lighter, just a spark. If I spark it, if it's dry enough and I spark it, what's going to happen?
It's going to catch on fire.
It's going to catch on fire. We call that spark activation energy. We call that spark activation energy. The energy for the reaction to occur. It is? Okay, so we'll continue this tomorrow. Great job today. Thank you so much. I owe all of you. All chemical reactions have that activation energy requirement. Some are big, some are small. In your body, you have and I don't know the number, but a lot of chemical reactions, a tremendous amount of chemical reactions happening in over 3 trillion cells all the time. You can't just go, well, it's hot, it's a day 102 degrees outside, 105 degrees outside. We go to the desert, we take a field trip to the desert, and now all of a sudden it's so hot, we just what? All the reactions happen at the same time? That's not a way to have life, right? You can't regulate life that way. So almost all the chemical reactions that happen in your body happen with an amount of activation energy that you can't find normally. Normally, in order to make a protein or to break one up at a body that's at 97 point something degrees or 96 point something degrees, this chemical reaction will not work. They do not work until your body decides, your cells decide to make a these chemical reactions in your body are here. They're not going to work unless your body makes well, we said enzymes. Makes the protein called an enzyme, and when you have the enzyme present, when that enzyme is present, you lower the activation energy. Now the amount of heat that's in your body is enough to have the chemical reaction go. So you want to stop the reaction, what do?
Stop the enzymes.
That's right. Get rid of the enzyme. Block it, stop it, turn it off. That goes to enzyme regulation, which we're going to get into next week. Enzyme regulation. Thank you. So in your body, that's what happens. Are we all clear on that? So that's how enzymes work. All right. So now let's go back to the first step in ELISA. I want to talk a little bit about some of the word antigen. Again, remember when we talk about ELISAs, you need surface proteins and antibodies. So whether you're talking about COVID, which looks like this, COVID, I don't know if you've seen it, but I've seen pictures of it done in an electron microscope, like the one Mr. Krishnan had in his room. These are surface proteins. And these surface proteins, what they do is we call these antigens. This is what the antibody, if we drew an antibody.
Ain't that what it recognizes?
That's right. That's what it recognizes. So this antibody recognizes this cell surface protein, this antigen. The antibody recognizes the antigen. Is that clear? On the outside of the cell, exactly. In this case, it's a virus. It's not a cell, but it could be a bacteria that also has these proteins that stick out of the cell. These antigens. Now, in tests, we don't put the COVID virus in your test, right? You don't want to send the virus. So we separate the surface proteins. We just collect the proteins. So this is what's in the test, the antigens. Where did we get the antigens? From the virus. Or in this case, if we're going to use the bacteria shape, antigens, or you can say, this is obviously exaggerated. I hope you don't think the bacteria looks like this. Or we could take the antigens from the bacteria. Again, we separate them using biochemistry. We can throw them in a centrifuge, break the bacteria up and or virus up with soap. Sometimes you even throw tissue into a blender, but you separate them, you break up the cells, you run them in a chromatography, some type of chromatography, you separate them, and you get this little container of antigens. The surface protein. These are very specific. This surface protein is only made by COVID. This surface protein is only made, let me pick a bacteria, E. coli.
So, is that how they remember? Because I remember watching the video, and it was like, the things in the body remember, like which type.
That's a really, really great question. What it is that the immune system is complicated. There's several different kinds of cells, right? One type of cell that's in your body is called a memory cell.
Yeah.
And the memory cells remember the antigens they've been exposed to. Now, some of them will remember. Some proteins are more easily remembered. For instance, measles. the antigen for measles, is remembered for a very long time. Because measles doesn't mutate that quickly. Now, so the measles from 20 years ago is going to have the same antigens as the measles from today. However, the flu, things like COVID and the flu, they mutate really quickly. By the way, remember, COVID is a type of flu. So flu and COVID, they mutate very quickly. So every year we have to have a new set of antibodies, a new vaccine that exposes your blood to the antigen. Because that's what we're doing. That's what a vaccine is. What a vaccine is we're taking these proteins and we're putting them in a container. Usually it's not a big container. These are all very small amounts. But we grow the bacteria or the virus in an egg, usually, because it's a perfect environment to grow it. Then we get a bunch of them, we break it up using chemistry, and we separate the antigens. Then we take these antigens and we inject you with the antigens. So you're never injected with flu. Originally, the very first vaccines, back in the days of the Revolutionary War, the vaccines for smallpox were actual virus. So in the case of smallpox, they would have smallpox parties, where pox means a boil, a pussy little filled on your skin. And they would people would get together, they'd bring one kid or adult that was sick, they would pop their boil with a needle and then smear it in there, get the pus, and stick somebody else. It's nasty, but it makes sense. Because in that boil, in that pussy capsule, right, is virus that's been attacked by the white blood cells. So those viruses are weakened. They're broken up. Just not completely. There's still live ones in there. But there's a lot of antigen, only a small number of actual intact virus. So by injecting you with it, you had less of a reaction. And your body was able to make antibodies and to remember that antigen. And next time you were exposed to it, your body said, and attacked right away. So you never got sick. So it worked. Unfortunately, a lot of people got sick too. But that's because it was a live virus. Today, we don't do that. You get injected with an antigen, okay, which is just the protein on the outside of the virus or the bacteria. Now, that said, I think that should be clear. What was I going to say? Yeah. Can't think of anything else. All right. So that's it for antigens. That's where that idea of antigen comes into. So one of the first steps that we're going to do when we're doing this ELISA is that we're going to have to create a buffer. We're not going to create it. It's coming. It should be in the supply list. The buffer is going to be somewhere around a pH of 9.6. Now, what is a buffer? A buffer is a solution, usually a weak acid or a weak base, what that means is it doesn't react easily. In this case, usually we use a bicarbonate buffer. It's a carbonate bicarbonate buffer. I'm not going to get into the chemistry of it, but bottom line is it keeps the pH around 9.6. So it's a base. Bicarbonate is a base, by the way. It's not an acid. It's a weak base, all right? Which means it takes a lot to react. So you can add a lot of hydrogen ions, and it's going to always end up being back to 9.6. You have to have an excessive amount of acid to get it to actually change pH. Let me pause this for a second. So a pH, so the buffer stays at, it's going to keep that solution at a 9.6. And then we're going to take them, we have these things, we have these. They're wells. They're plastic plates. You've seen them. You've worked with them in the past probably. But I know that in my class, in biology, I know you've seen them. There's a bunch of little, well, we call them wells. Do you understand what I mean by wells? Right? There's little spaces in here. Does that make sense? So when you put a solution in here, little drops, you're going to put drops. It's going to fill the well. Am I making sense to you? Yeah. So these wells hold liquids. They're made of plastic. We call this whole thing a well plate. When I give you the procedures, you're going to read this. And you're going to see well plate. If you go do research in an intern in a lab, you're going to see the term well plate. All right. So you're going to incubate in the plate overnight at about 4 degrees Celsius. It's above freezing. Or you can incubate it for 37 degrees Celsius for 2 hours. So this is overnight. We're probably going to do the 2-hour one, 37 degrees. Now, this is going to be important when we do this step, because at this step, what we're doing is we're creating a coating that we're going to make so we can make the test, the antigen, stick to it. So we have to make sure that we get enough of this on there. So it's either going to be overnight at 4 degrees or 37 degrees for 2 hours. And again, we're going to probably do the 2-hour one. So at this point, the temperature is important because you want to keep the shape. The shape is important. And we all know that if you get it too hot, it's going to lose, it's going to denature. And if it's too cold, you won't get a reaction. So we have to time this correctly. So if this is us making sure that we create the antibodies correctly. We're going to actually create these antibodies. So we're going to incubate them. And if we don't do the right temperature, it's not going to work correctly. And we don't want the antibodies to denature, because this shape that I've drawn on the board that you've seen in your book, if you've read your book. I like how you say book. What, we have a book? What?
I didn't mean to say it like that.
So we have a shape like this, and we have another piece of the antibodies like so. And this is the active site, or this is the site right here that's going to stick. If you mess with this shape, then they won't match the antigen. The antigen and antibody match because of their shapes. Just like the enzyme. We're talking about shapes here. So it's important that we keep everything in line. So not just the pH, because that can change the shape. That could denature a protein. But also, not only the pH, but also the temperature. Also the temperature. Okay. So when we look at all right. So again, we don't want to destabilize these proteins. So we need to have it at 9.6. So we keep it at that pH using a buffer. We keep the temperature because we don't want to destabilize these antibodies shapes. Is that making sense to everybody?
Yes.
All right. So what is it we're going to do in general? We're going to take these antibodies that we're going to create. We're going to make sure we're incubating them in a Petri dish. And tomorrow I'm going to, you know what? What time is it? How much time do we have left in class? 2 minutes? I'm not going to get into the rest of this. Tomorrow I'm going to let you watch a video and you'll see the actual process in whole. I want to introduce you to an outline of ELISA. Again,
Isn't it spelled with a Z?
no. It's enzyme linked immuno. What is it? Immuno. That's right. Immunosorbent with the immuno-sorbent S assay. Thank you. So ELISA is basically you have this well plate. It's called a well plate. And you know what a well is, right? Okay, so it's a plastic plate that has little wells in it. We have a bunch. You've seen them last year, or I'm sorry, it might have been last year for some of you, but in 9th grade we had them in the classroom. There's some that have a few, some that have many. And inside the wells you can put liquids. All right. So we call this a well plate. And you're going to see it when you read the actual lab protocol. And these are your samples. Now, the first thing we got to do is I'm going to do a close-up of one of these wells, if you don't mind. A cross-section of one of these wells, if we see something like this, okay? And what we want to do in the end, by the end of this, what we want is the bottom part of the part that doesn't stick to the antigen of the antibody to be sticking to the plastic of the well plate. So we want it to look like this. We want that well plate to be covered in it. Now, if you did a COVID test at home, or if you've seen a COVID test done, then you should know that that little indicator or a pregnancy test works the same way. They have the antibodies sitting on a sheet, a very thin sheet of paper, and they're just sitting there like that. If the antigen comes in and it attaches, it changes color. So there you have your antibodies. They're sitting in the well plate. So our first step, the first thing we're gonna do is we're gonna coat our well plates with antibodies. So they're gonna give us antibodies. We're gonna have antibodies and this micro well plate. It's actually a micro well plate because that's mu for micro. Mu is a Greek letter. Those of you that are going to swear to fraternity, Greek fraternity, you should know your Greek letters. So, in any case, the wells are coated with these antibodies. You'll be using probably a 96 well plate, 96 well, so there'll be 96 wells, and it's called a microplate. That's a symbol for micro. And we're going to incubate it. We're going to put a drop in there, swish it around, and then we're going to let it sit. We're either going to let it sit at about 4 degrees overnight, or we're gonna set it. Another possibility is raise it to 37 degrees for a couple hours. We're probably going to do overnight. Actually, I was thinking about it. It's probably going to get us a better result if we just let it happen slowly. And the objective is to let the liquid evaporate and have slowly, and let the non-polar region of the antibody bond to the side of the well plate. It's very delicate. So when you're handling them, when you do the lab, you have to be very, very careful. Because you don't want to after spending 24 hours or overnight sitting in a 3 degree Celsius, it's just a little bit above, it's in a refrigerator, letting it dry out slowly. You want them all to line up nicely so that the lab works. So then you rinse carefully. You have to rinse carefully. So you're gonna take some rinse, and we'll talk about what that rinse is made of. And you're gonna rinse off any excess stuff that's in there. So only the ones that are stuck on the side of the plate. But now you got a problem. Now you got these plastic spaces here. They're hydrophobic because it's plastic. Plastic is hydrophobic. That's why we use it to contain water, right? Because water doesn't stick to it. Does that make sense? That's why paper cups don't work as well as plastic cups. So in any case, they're hydrophobic. So we got to make sure that we don't get any sticking going on here during our test. So what we do, we add a blocking buffer. It's called a blocking buffer. That's the second step. A blocking buffer is made of stuff like, non-fat dry milk, or it might be a casein. You probably have heard of casein used in our foods, but something that's going to stick to the plastic and not allow any more hydrophobic interactions happening. So you put that buffer in here. It's usually has a buffer is a material that stays a certain pH. So it's usually like 9.4. It's a basic pH. And that way you don't get any more sticking in places where you don't have antibodies. So the only thing that's going to stick now is the stuff that sticks to the antibodies. Oh, is it time to go? Thank you. Sorry about that. I get involved in this and I lose track of time. All right, I'll take attendance. Please clean up and put these. Just put the chairs up, and as soon as all the chairs are up, you can leave. So now we're ready in the third step to actually add the sample. Step 3 is we're going to add the sample. In the case that we're just using here, it's that the antigen that we were talking about earlier, that spike protein that we looked at earlier on COVID. And we have a sample from the rabbit. The rabbit made us antibodies, and from the egg and multiplying the virus inside an egg and isolating it using biochemistry, we were able to get the antigen in a little tube or in a container. So now we're going to add this container. Now, because this antibody matches this antigen, if you put it in there, if what you're adding has that in there, has the antigen in there, it will stick. Does that make sense? So if we took a sample from your nose, your snot from your nose, which snot is a protein, by the way. Mucus, the real word for it is mucus. And if we take the mucus from your nose, this is what we do for COVID tests, right? We take a Q-tip, a long Q-tip, we swipe it in there. We get that mucus and we put it in this well, right? Then, huh? Oh, is it time to go? Oh, I forgot. Thank you. Have a good one, guys.